All right, this evening's lecture, Digital Fossils. Well, apparently it's nothing to do with old bits of hardware in your basement, the loft or corner of your study. It's much more interesting than that. I've been given an awful lot of words, but I've also been given instructions not to read it all. I need to pick out some highlights. But I'm just entering the stage of life where I need reading glasses, so it's not.
So I've got some in my bag.
not as easy as it might be. But this is Professor Emily Rayfield, our speaker for tonight. She received her undergraduate degree in biological sciences from the University of Oxford, PhD in earth sciences from the University of Cambridge, and postdoctoral research fellowship positions Oxford, Cambridge, and the Natural History Museum in London. And in 2005, took up a faculty position in the School of Earth Sciences, University of Bristol, where she has remained since. Recently received the Gabor Medal for pioneering a new cross-disciplinary era of engineering-informed computational paleobiology. I think that's as impressive as it is difficult to say. Emily's research focuses on how skeletal mechanics influences morphological evolution and the relationship between form and function in biology. Yes. And I'm assuming we're going to be seeing more of that this evening. We are. So to wrap up with the mini words, it also says here that Professor Rayfield's highly interdisciplinary approach, and again, I think that's what we'll be seeing this evening in the talk, has seen her at the forefront of her discipline, developing novel and creative approaches to study organizational function. Not organizational function, that's what I do at work, sorry. Organismal function of organisms and their evolution. Right, and on that note, I think I'll shut up and hand over to Professor Rayfield to give us our talk this evening. Thank you.
Great, thank you very much. And thanks very much for having me here this evening to speak to you. And thanks for a lovely dinner as well too that I've been treated to before talking to you. Yeah, so I'm going to talk to you today about particularly how we can use X-rays and other methods to understand fossils, so extinct animals, particularly how they lived and interacted with their environment. And also as well with a focus on, we're familiar, I think, a lot more now with reconstructions of fossil animals and extinct environments in TV programs and in films. And part of the lecture as well is going to focus on really, how much do we know about what we see and how far can we actually understand what's happening in these documentaries, what's based on fact, what's based on estimation, and what's more fanciful. And so I'm going to start with a TV documentary that I was very fortunate to be involved with, and we're going to use this as part of the focus for the first part of the talk. So you can sit back and relax for a minute or so and watch this.
I'm on the south coast of England, in what is known as the Jurassic Coast. 150 million years ago, the land was ruled by dinosaurs. But the oceans were dominated by a mysterious sea monster. Now, fossil experts are working to unearth this extraordinary find. No one's ever done this before, extracting a giant skull halfway down a cliff face. This colossal creature is called a pliosaur. But little is known about these prehistoric giants. What else? Terrifying thing. Together with a team of scientists, we will use new technology to discover more about this enormous marine reptile.
I have no doubt that this pliosaur was the T-Rex of the seas.
Comparing ancient animals alive today, we can deduce a great deal about their shapes.
This pliosaur has a bite force that's about twice the great white shark.
Could this be the greatest aquatic predator that ever lived?
So that was a TV show that, as you obviously saw, I was very fortunate to be involved with. It came out on New Year's Day, the start of this year. And there's me at the end saying about this fossil find that here, it had a bite force twice the size of that of a great white shark. And obviously, you know, I'm saying that on TV, you know, part of the talk today is going to be like, how on earth can we actually make those kinds of statements? What is it actually based on? How can we understand, you know, not only, you know, what these animals look like, but how they were feeding, biting to such great bite forces like that. So this is the specimen itself that the documentary was based on. It's a really beautiful fossil. It's an animal called a pliosaur. It's a marine reptile that's around in the Mesozoic. That's a period from, this particular animal was from the Jurassic. So we're looking at about maybe 150 million years old. This is a fantastic specimen as well. It's really well preserved. It's also got minimal distortion as well too. You can see the back of its skull there. It's just a little bit twisted to the side. The specimen itself is on display in the Etches collection. This is Steve Etches himself here, who over many years has collected remarkable fossils from the Dorset coast and has now got funding to put them all in a dedicated museum. You can find this museum in Kimmeridge. So we were fortunate enough to study this specimen as well too. So how do we go from a fossil that looks like that to these kind of CGI reconstructions here and the statements that I'm making on national TV? So I think we'll start off though with thinking a little bit just about what evidence do we have for how animals actually of the past interacted with their environment. A lot of my work focuses on the skull. It focuses on how animals fed, what they fed on, that kind of thing. And one of the best examples of what an animal might have been feeding on are actually fossilized preserved gut contents. They're kind of smoking gum for at least what was the last meal of an animal before it was actually died and was fossilized. So this is a specimen of a dinosaur. It's from the Cretaceous period of China. Really fantastically preserved fossils in this lake biota. That preserve not only the bones, but often soft tissues as well too. This animal here, here's an inset of its, inside its rib cage here, you can see the ribs and you can see this kind of brown mass in the middle. And then there's a couple of long bones here, which look a bit out of place. Now, if we look at these down a microscope, what we actually find out is this brown mass is the remains of partially digested feathers. The reason where they're sitting in the rib cage there suggests that these weren't really washed in, that they were actually part of the animal's last meal. And here's a little reconstruction of this animal eating a bird. These two bones here, though these long bones here, now we're actually fortunate that we can actually identify what they belong to. So sometimes fossils have what we call diagnostic features, which are really distinctive, and we can tell just from a few isolated bones what animal those bones belong to. And in this case, we can tell these bones belong to another dinosaur. And they really have this little, and they're the hind limb bones as well, some of the long bones of the lower leg. And here's also a little reconstruction then of our animal here with the hind limb of a dinosaur in its mouth as well too. So other good evidence as well too. This is a fossil called Microraptor. It's from the same age and the same deposits as the previous animal. Despite having wings, or 4 wings in this example, it actually is still a dinosaur. We know this because its skeleton is actually still shaped. It has those diagnostic features of what we will call a non-avian dinosaur, a dinosaur that is not a bird. And again, this is preserved here in these flat specimens. Whoops, And I was explaining over dinner actually, there are often hundreds, if not thousands of some of these specimens actually found. And occasionally you also find gut contents. So in the top there, where we see that yellow box, if we zoom in to part B here at the bottom, you can see that there are these longer bones here which represent the animal's ribs. But inside there is these spindly structures. And if again, we look at those in great detail, we can see they're the bones of a foot. And they're actually the bones of a mammal foot. And we know that because mammals have got really distinctive ankle bones. And you can see those ankle bones preserved here. So this is a dinosaur that is eating a mammal, and the dinosaur itself looks like a bird. And in this little cartoon here, we can see the animal with a little mammal paw in its mouth. So good evidence there for that these animals form part of the diet of other animals. So other things as well too that give us clues are when we find things like bite marks, occasionally we find teeth embedded in the bones of what was the animal's prey. In this case, the pencil is pointing to a tooth that's been embedded in the backbone of a herbivorous dinosaur. But in this case, the animal got away because the bone has actually grown around the tooth. And then eventually the animal's died, and we've got that preserved in the fossil. And then in other examples, we find bite marks and teeth also embedded in bone as well too. So we can see that there's a predator-prey relationship going on with it. Another good example of what animals were eating comes from coprolites, fossilized feces, basically. And so this is a very famous coprolite that was found in the latest Cretaceous of the United States. And those black and white bars on the scale bar represent a centimetre each. So you can see this is a pretty massive structure. It's been sectioned as well too. If we look inside, it's chock full of shattered pieces of bone. So we know two things. One is that, well, three things. One, it was produced by a very large animal, an animal that ate other animals and was powerful enough to shatter its bones. And also, because these bones are fragmented and not very worn or etched by stomach acid, they probably pass through the gut quite quickly as well. So it's got quite rapid gut movement times. And so the size of this specimen, the area it's found in, leads to a large meat-eating dinosaur. And the most common fossil that we find in those environments is Tyrannosaurus rex, the tyrant lizard king. And hence, when the paper that described this fossil produced, it was known as a king-size theropod, the theropod dinosaur being the meat-eating dinosaurs. So we can use other general evidence as well to link animal skeletons to the things that they fed on. A great example, an obvious example, are teeth. So at the top here, we've got the skull of a modern Komodo dragon. It's got these really characteristic, sharp, recurved teeth, and we find these teeth in many other meat-eating animals, lizards, other types of animals as well. And we find these shapes of teeth in many of the meat-eating dinosaurs and other fossil animals that we see in the fossil record. On the bottom is an iguana, a living animal as well too, and it's got these really distinctive leaf-shaped teeth. And these leaf-shaped teeth we find repeated numerous times in the fossil record in what we think are herbivorous dinosaurs and in other animals as well too. And this seems to be linked with these animals eating plants. So we can make these broad-scale comparisons between the shape of the teeth and the type of foodstuffs that the animals are eating. But then, you know, how do we go then from that to things like me? 5 foot, it's about twice the great white shark. Twice the great white shark. So there are a number of things we can do. So one of the things that we do in my lab and that other people do as well is look to living relatives of fossil animals for clues. And in this case, what we're trying to understand is the arrangement and the type of muscles in the skull that help close the jaw. So one of the things we can do is dissect living animals. Crocodiles, alligators, are a pretty good analog for fossil animals like dinosaurs. We think that they have a pretty similar arrangement of jaw closing muscles in the back of their head. And what we're doing here on this picture is dissecting a large alligator. At the moment, this is just the head you can see here, but it was actually the whole specimen that we had. And we were doing this at the Royal Vet College, just north of London, because they have facilities there to actually dissect very large animals. And we're just removing the skin there, which is really hard. And underneath it, you can see tissue, and then in yellow and pink are a lot of the muscles. Those big muscles that are bulging out behind the head are ones which either open the jaws or close the jaws at the back. So once we go through the dissection, we can then produce these maps of where these muscles actually attach and where in the skull and then the lower jaw. And we can get a sense of the spatial arrangement of muscles as well, and also how big those muscles actually were too. Once we've got that information, we can then produce these maps of where the muscles attached in the skull and in the lower jaws. So we obviously, with living animals, we can look at the animals, we can observe them, we can dissect them. What about fossils? Well, fortunately, the bones of fossils leave us with some clues. So in the bottom left-hand corner there is actually a fossil crocodile. The fossil crocodile skulls are pretty similar in shape to the living crocodiles. And as far as we can tell, they probably had similar types of muscles to close the jaws. So those different coloured regions there represent where we think different muscle groups actually attach to the skull. The muscles that are in croc skulls, in our skulls, don't form one kind of single muscle mass. There are different muscle bodies that will contract at different times and pull different parts of the skull and jaws. Unfortunately as well too, the bones leave us other clues too. So where muscles attach, we often find either smooth surfaces or we find ridges which border the edge of where muscle attachments were, or perhaps tendons were attaching into these roughened surfaces. So we study the fossils in detail, we can get these little clues of where muscles might have been attaching. And then we can produce these muscle maps in fossil animals like we did for the living animals as well. And so at the bottom right here is a muscle map that Andre, who works in my lab, who you saw in the footage just before me, has produced for a Tyrannosaurus rex skull. And so these are not definitive arrangements of where we think the muscles are, but they're our best estimates based on studying living animals, based on studying the fossils themselves, and trying to deduce where we think some of these muscles might have attached. So then we're going to have our first kind of foray now into how X-rays then can help us understand the muscles even more. Because what we've got now is an idea on the skulls of where the muscles might attach. But clearly those muscles have got to fill gaps within the skull. We need to know their 3D kind of arrangement. And one of the ways in which we can do this is use X-rays. So X-rays, first discovered by Wilhelm Röntgen, who was a Dutch-German scientist working in Germany, who discovered the fact that electromagnetic radiation was being produced within his lab, which actually was producing these, passing through objects and producing these images. And here on the right is an image he took of his wife's hand. He also took images of his own hand as well. Both of these are quite kind of iconic when people are discussing the earliest X-rays. And all of us are familiar with X-rays. We have had them at many times during our lives. In dentistry, X-rays are used via an X-ray tube here, which produces the X-ray source, which then takes images of our teeth and jaws. We also, you'll be familiar with CT scans or CAT scans. The difference here is if you've ever had one and you lie on a flat bed and the X-ray source and the detector moves around you. And as it does one circle, it produces these X-ray images, which then can be used as 2D slices through the body that you can see here, or when numerous slices are computationally stitched together to produce these 3D maps of body organs and parts of the skeleton, for example. So we can use these exact same principles to X-ray animals. Now, typically in our lab, we have used these medical scanners, but we tend not to use them for a couple of reasons. One is that they're great for specimens that are really big, but the resolution that you get is not very high. So each rotation is like at best usually like a millimeter or a little bit less than a millimeter. And some of the features that we want to resolve are a lower resolution than that. And also as well, the energy that these X-rays produce, obviously because they're X-raying us, is not very high. And sometimes when we've got really dense material, we need to use higher energy to actually resolve some of the skeleton and some of the tissues inside. So we use micro CT scanning, the micro bit, not because the machine is smaller, the machine is pretty big. This is the machine that we have here in Bristol. It's in a lead-lined box. It's about 2 meters by 3 meters in size. But the micro refers to the resolution of the scans that we can get. We can go down to, perhaps, just a few thousandths of a millimeter in resolution. They're really, really high resolution. It depends on the size of the specimen you put in and how far that specimen is from the detector. And in this case, here's on the right, this is Liz, who is our Liz is actually quite small, but she is, nonetheless, the chamber is pretty big that we have for our machine. And on the left of Liz is the X-ray source. And in this case, rather than the CAT scanner where the X-ray moves around, we would put our specimen on that detector plate, which is Liz is sitting in front of, fix the specimen really carefully, as well, especially if it's a rare fossil. And then the plate itself rotates around. The X-ray source remains fixed. And then behind that plate is the detector. So the X-rays are passing through our specimen. Depending on the density of the specimen, they're attenuating. And then we're getting an image on the detector plate on the back. We do this multiple times. We may rotate the specimen 1,200, 3,000 times or whatever in really, really tiny increments and build up a series of these slices and get these really high-res scans. So this is really, really great for looking at tissue. So this on the top here represents in Bristol Zoo, before they moved, they used to, I don't know if you fed the lorikeets at the zoo. You could go and feed one of the lorikeets, they give you a little pot of stuff and you could feed them. And sadly, one of the lorikeets passed away and we were able to use the specimen for a project that actually a student of mine was looking at parrot skulls, because parrot skulls are pretty ridiculous because they've got really thin bone, but they can, if you ever had a pet parrot, they can produce really, really high bite forces, but they've got ridiculously thin bones and they've got a massive hinge in the middle of their head as well. So we were really interested in looking at how parrots worked. Anyway, so this is essentially the procedure. So we got the specimen of this in the next slide along here in the X-ray image. The specimen is upside down in a tube and it's on our rotation plate. And this is the first kind of X-ray image that we would get. We take a series of these as the specimen rotates around, and then that builds up these 2D slices like we can see here. And what we're looking at here is like a cross section through the head. So at the back, this is the cavity where the brain would be. And then this is the front of the jaw and the beak here. And then by using computer software, we can stitch all those 2D slices together and produce these 3D images here. And you can see the resolution of this. I mean, the heads of these animals are really tiny, but we can pick out the beak really clearly, the bones of the eye here, the sclerotic ring, which sits in the eyeball, and then also things like the neck here and the trachea and so forth. However, when we use fossils, there's an extra complexity because the fossils have been mineralized. And so they've been petrified, they've been turned to rock. And so we need to use much higher energy to actually penetrate through the specimens. And that means often the scans that are not as pretty as the ones that we see here. So this is another specimen that we did in Bristol. This is a 220 million year old sort of fossil proto-mammal. That's it down there on the left, it's from Brazil. These animals are really important, and I'm going to talk a little bit more about mammals in a minute because they sit just outside the origin of mammals. So they can tell us a lot about the evolution of mammals and when key mammalian features actually evolved. So this is a collaboration that we have in Bristol with our colleagues who are in Brazil and Argentina, and we're fortunate to be able to scan these really lovely fossils in Bristol. You can see this one's been really nicely preserved, and the bones this is really unusual, the mode of preservation as well, because normally, the bones that you would find are kind of a black or a grey or a browny kind of colour. In this deposit, they preserve in this beautiful white colour here. So they almost look like they're still living modern animals. But you can see behind this orangey stuff is the red sandstone matrix, the rock that they're actually preserved in. So here's our X-ray of this animal. Again, it's in a little, this thing is tiny, it's about 2 centimeters. It's in a tube, it's on our rotation plate going around, and we've got some X-ray images, and we got those and thought, oh, they look pretty good. And this is what you get out here. And this actually is a pretty good scan of a fossil animal. You can see the white, or the lighter grey here is the bone, and the darker kind of crumbly material is the rock. So we use the software in the lab then to digitally extract the rock from the bone. And we produce these 3D models here, like the one you can see. So we can use these just to understand the anatomy better because we can see inside the fossil. But we can also use these scans to then look at kind of how the jaws might have worked and do further analysis on them. I'll talk a bit more about that in the talk. So that's a little foray into like X-rays and how we use them. So I'm going to come back around now to thinking about mussels and making estimates about forces. So we can get information on how big mussels are by using X-rays and CT scans of living animals, so to get those 3D information. And this is actually not a picture of a skull here. This is actually a CT reconstruction of the bones that are inside the head of this specimen here. And this has also been digitally recreated from our scans. It's a grey short-tailed opossum. And the way that we've got such nice resolution for the soft tissues is we use a method called contrast-enhanced staining, which is a fancy way of basically saying we take the specimen and we dunk it in a solution that then leaches into the tissues and makes them attenuate the X-rays better. So this is what's currently in the lab at the moment. This is a gar, a fish with an extremely long snout. It's in a solution of potassium iodide, and the iodine is leaching into the tissues just by diffusion, but it's going to increase the contrast of the soft tissues. Normally when we scan stuff, you get nice pictures of the bone, but you can't see the soft tissues. So when we did this for our opossum, you can see here on the left and the right the difference it makes, and this isn't even completely finished yet. So this is without staining. We just took the skull, put it in the scanner. Nice images of the bones, but everything else is just grey around the outside. After it's been soaked in some of the contrast-enhanced stain for a while, we start to pick out these mussels really nicely in the scan. Give this more time as well too, and the stain leaches in even further, and we get really nice reconstructions of the mussels. So we can use the same software then that we use to create those 3D images of bones to create 3D images of mussels as well too. And indeed, that's what we did for this project here. So we can build these digital maps of all the mussel bodies in the skull based on what we see in the scan. So this gives us a clue not only where those mussels attach, but also how big they are. And that's really important to know because we want to actually make an estimate of how much force those mussels can generate and how hard the animal can bite. So a little foray then into thinking about mussels. I'm absolutely sure some of you in the audience are medics, dentists, nurses, whatever, and this might be familiar to you. But you can see I've taken this image from a medical textbook. So mussels are kind of constructed in really very similar ways. And as far as we know, fossil mussels looked exactly the same as this, because if we look at living representatives of mammals, dinosaurs, birds, their mussels are all built in the same way, which means that probably all those fossil animals have mussels which were really similar as well. So mussels have got long fibers which are attached at either end, either to tendons or straight into the bones themselves. And the force that mussels can generate depends not on how long they are, but what's called their cross-sectional area. And that's basically the area of the mussel fibers. When you've got a mussel that, like we have on the left here, that's parallel, like your hamstrings, those fibers just run in long directions along, often overlapping like that. Sometimes though, in other mussels, like the quads here, you've got a tendon in the middle and the fibers branching off from the side. So if we can get an estimate on the cross-sectional area of the fibers, we can then work out how much force that muscle can generate. So we use these kind of calculations here. I'll just kind of walk you through them so you can understand what measurements we actually need. So we calculate something called the PCSA, which is the physiological cross-sectional area, which is the area of the fibers. And to do this, you can either use the mass of the muscle, so literally you dissect it and you put it on a balance and you weigh it. And we can then take that and divide it by the density of the muscle and the length of all these individual muscle fibers. Muscle density is a fairly standard value. It's pretty much just a bit more dense than water, like water with a bit of protein in it, basically. If we could also use, though, the volume of muscle and divide that by the length of the fibers. So if we haven't got anything to dissect, what we could do is take one of our digital reconstructions and use the computer to tell us the volume of those muscles. When we have the pennate muscles with the tendon, it's the same principle, but we just take into account how far the fibers are angled off that central tendon. Once we have that value there, we multiply the cross-sectional area by muscle stress, so the force per unit area. And again, this is what we know from living animals is pretty conserved in terms of values. You know, muscles tend to function in the same way. They have their overlapping units, they contract over each other, they release, they move apart. And so we can use that then to work out how much force the muscle can actually generate. So there we have our muscle force values. Now we're nearly on the way to thinking about how much bite force an animal could generate, even how fast an animal could run if we understand the leg muscles. So I'm going to have a little detour to show you then how we did this in a really unusual fossil. So this is another dinosaur, and it's an absolutely bizarre animal. So this is an animal called Erlikosaurus. It's again from the Cretaceous. It's a member of a group called the Therizinosaurs. And remarkably, in the Cretaceous, these crazy animals were actually globally quite widespread. We find their remains in North America, and we find them in Asia as well too. They were kind of large-bellied, squat-legged. Distinctively, they had these arms with ridiculously long claws on the edge of them. But you would imagine like that, you know, imagine you can't see the skull. This was a terrifying animal, but then on the top of its neck, it's got this fairly puny little skull with tiny teeth and it ends in a beak. And so the general consensus is the teeth that we see in the jaws as well are those leaf-shaped teeth that we see in iguanas and other animals that eat plants. So we think this animal was a plant eater. We're not entirely sure what the claws do. I've had a student looking at how strong the claws are, and he thinks they're not very strong at all. That they might be used for grasping plants, or they might even be used, he thinks, as some kind of signaling mechanism, you know, in a similar way to deer use antlers and things like that. I think the general consensus is we really don't know what they're actually used for. But we were interested in looking at the skull of this animal, and we were very lucky. A few years ago that this chap here on the bottom right, Pele Oltangro, who sadly has passed away now, but is from the museum in Ulaanbaatar, where the specimen in Mongolia was actually kept. He came on a trip to the UK, partly funded by the Geologists Association, and with him brought this skull. And so while it was in the UK, we were all like, oh my God, we really need to study this. This is amazing. It's like a once in a lifetime opportunity. So we were fortunate to get the skull CT scanned, and here we are actually scanning it here using one of these micro CT scanners. Pearl is holding the skull there. You can see how small it is, and you can see there are these puny little teeth, and then there's at the end of the jaws here, there's no teeth at all. If you turn it upside down, there's just an edge here with all these little holes on, which is classic what you see in animals like turtles and other extinct animals that have got a beak. So we were able to use this skull as well. It's nearly complete. It's really beautifully preserved. And Stefan here was my postdoc and PhD student at the time, and he now works in Birmingham, was able to literally digitally fill in the gaps here and create this really nice reconstruction. And, you know, with this, we were able then to, you know, do little things with the CT scans, but we were also able to produce these muscle maps to think about where the muscles in the skull might have sat. And was this animal's skull and jaws really as puny as we thought it was? So Stefan produced these really nice muscle maps. Again, all the evidence suggests the muscles were really similar in arrangement to what we see in rocks and living alligators and things. So from that, we were able to create these kind of tubes where we join the muscles up together. And then Stefan was able to flesh out the muscles to think about spatially how they fitted within the skull itself. And then produce these kind of 3D maps of how much of his best estimate, shall we say, of how big he thought these muscles actually were. We can then use the measurements that I was telling you about before to get an estimate of how much force muscles of that size might generate. So the next thing we think of is we then want to convert those forces the muscles are generating when they're contracting at the back of the skull to a bite force that's happening at the front of the jaws. And we're fortunate because jaws basically work like levers. So they have a joint at the back and they have a pulley, an in force, which is the muscles pulling, and then they essentially have an out force at the tips or wherever it is the animal's biting. So we can use kind of simple lever mechanics essentially to say, you know, we've got the hinge here, we've got fulcrum here, we've got a force pulling here, what's the out force here when the animal's biting? And that's what we did for this animal based on our estimates of how big its muscles were. And what we found was at the back of the skull, the animal's biting with a force of around 100 newtons. This is very variable because there's lots of different things you can do to think about how big the fibers were and stuff like that. But to put that into context, this is about how hard a 6 to 8-year-old child would bite at the back of its jaws. In the middle of its jaws, it's biting at about 75 newtons. Because of the way the lever's working, the further you move forward, the lower that output force actually is. And so that's about the same as a kind of pre-teen, early teens could bite at the middle of their jaws. And you see the growth stage also we're seeing here as well, using my children as an example. And then the front of the jaws, this animal's biting at about 50 newtons, which is equivalent to what a teenager can bite at the front of its jaws here. So humans themselves, actually, adult humans, like George pretty much is here at the bottom, can actually generate really high bite forces. At the backs of their jaws, they can get up to maximum records, and it's been about, you know, almost like 20 times this value here. And that's because we as humans have got really short faces. So the leverage of our muscles is much more advantageous. This animal here, the longer jaws means that as we go towards the front of the mouth, it's got a pretty puny bite force. So again, this ties in with this animal having, you know, fits in with this whole bizarre nature of this animal. Big animal, large claws, puny skull with puny bite force. So back to our pliosaur then, and this, you know, 2 times the bite force of a great white shark. So we can use similar principles here to look at how big we think those jaw closing muscles were, look at the leverage of the jaws, and then give us an estimation of how much force this animal was generating during biting. Now, this is not a 20 centimeter long weird dinosaur skull. This is a 2 meter long skull, and the jaw muscles are sitting in those big openings at the back of the skull. Now, actually, with this, we didn't use X-rays. This is a bit of a cheat, and that's simply because this skull is so huge and so dense as well, that any scanner we had, at least that was available to us in the UK, was not powerful enough to actually resolve any of the features. In the TV documentary, you could see there was some scans there, and that's because in Southampton, they have a really powerful scanner that's nearly twice as much energy as ours. There are very few of these in the UK. They were able to resolve the very front of the snout. But anything further back, it was simply too big and too dense to actually get anything out of it. And of course, this thing literally weighs a ton as well. So you have to try and get it on these machines. We tried to scan an ichthyosaur specimen for a previous documentary that's about the same size, and it was simply too heavy to go into any of the scanners that we were able to get hold of. It's it's simply not possible. There is a famous example of a T-Rex skull that was scanned at a scanning facility in the US, which belonged to Boeing, and that was big enough, could hold specimens that were heavy enough and had high enough energy to actually, for the X-ray beam to actually penetrate through. But what we're doing here is actually using what's called a surface scanner. So this is taking literally a series of kind of photographs of the surface of the specimen. And this is Andre here, you saw in the documentary. He's moving the scanner over the surface of the specimen. And here's Andre. Andre spent his PhD doing this, basically, in dinosaur skulls. And here he is in various institutions on cherry pickers and up ladders, basically creating these like 3D models using the surface features. Now that's great, but we don't get the internal detail from the scans. But what it does allow us to do is to build up these 3D surface models. And so we use these surface models then to estimate how big we thought those cavities were at the back of the skull. So in here, in those red areas, these were chock full of jaw closing muscles. And this is the estimation that we used to come up with this idea of 2 times the bite force of a great white shark. So this is just a snapshot of the calculations that we were doing. We basically had a whole series of different estimations based on different types of parameters. And our kind of best guess, most powerful bite was the one that was 2 times the estimated bite of a great white shark. So to give you some kind of context here, in terms of how hard a human can bite, the highest, most possible bite a human can have, the pliosaur was about 64, or kind of like the average bite of a human, shall we say. The pliosaur can bite 64 times more forcefully than that. And then here's a whole load of other estimates here. Here's our great white, here's our T-Rex, lions, hyenas, 2 of the largest bite forces that are produced in terrestrial animals. And then here's our giant alligator at the top here. And this is literally how you measure bite force in an alligator. So on the end of that metal pole there is a force transducer, a small tuning fork, which the animal, which is covered in rubber, the animal's biting into, and that's sending a signal back to the amplifier, which is recording the force of the bite. Greg Erickson there, the guy who's doing it, is getting a bit closer with his pole to try and estimate that bite. So that's how we can kind of come up with those measurements. They're broad brush estimates, but that's what they're actually based on. It's our understanding of living animals and our reconstructions of the muscles using those principles. So then in this next bit of the talk, I just want to talk a little bit and kind of switch back to thinking about mammals. We've looked at giant Mesozoic marine reptiles with massive bite forces. I'm now going to talk about tiny things that were around a little bit earlier than our pliosaur, but in the same kind of geological period. And so these are Jurassic fissure mammals. So what do I mean by this? So the Jurassic here in our geological timescale is a period, one of the 3 periods of the Mesozoic era. And you can see the Mesozoic runs from about 251 to about 65 million years ago. The end of the Mesozoic is the KT or KPG boundary, the asteroid impact, kill off the dinosaurs. So that's the time frame that we're actually looking at. And I particularly like this picture here, because this is a reconstruction of Jurassic Scarborough. I like this for a number of reasons. One, it's got fossils in. 2, I'm from North Yorkshire, and so this makes me smile when I see the Jurassic of Scarborough reconstructed with these large plant-eating sauropod dinosaurs with the long necks on one side, and there's a meat-eating dinosaur as well lurking around. And this was made by an artist who lives in Bristol, Bob Nichols, who does some great reconstructions of fossils and past environments. And it was made for the Rotunda Museum in Scarborough. I think it's not in there anymore, but a lot of it is now on display in the Yorkshire Museum in York as well too. And so this is a time that's very different, obviously, to now. The British Isles was much further south. The climate was very different. Obviously, the animals were completely different as well too, or were they? So we typically think of the Mesozoic as being like the age of reptiles or the age of dinosaurs, and indeed they were the dominant animals on the land. And things like pliosaurs were the dominant predators in the sea as well. But actually, for a large part of the Mesozoic, we also had mammals. And if you think about it in terms of the time, here's the asteroid impact 66 million years ago. Here's the Cenozoic period, typically thought of as like the age of mammals, which is everything from the impact to the present day. And this is when we have the radiation of all the modern groups of mammals we're familiar with. Prior to that, though, we actually have about 2/3 of mammalian history in the Mesozoic. And actually what we've got in the Cenozoic is only about 1/3 of that. And so living among the dinosaurs were all these different groups of what we would call Mesozoic mammals. And these represent mammals that kind of would look pretty much familiar to us today, so but as in a small kind of mammal type animals. But they actually represent not things like lions and tigers and whales and stuff, they came much later, but they represent little radiations of mammals, which then all went extinct either at or before the end Cretaceous extinction. But the point is that there were lots of mammals around at the same time as dinosaurs. And one of the things that we're really interested in, here's a reconstruction of a mammal and a dinosaur as well too. I was just hearing you had a talk about colours in dinosaurs perhaps recently, and that some of my colleagues in Bristol have been involved with, and you can see this image here has kind of got some of the gingery colouring in that animal as well too. So we're particularly interested in the origin of these groups of mammals. And one of the great places that we can look for these is actually quite local to us where we are now. Now, this isn't local, this is Malham Tarn, again in Yorkshire, but it represents a nice example of a limestone pavement here. And these are the kind of deposits that we're particularly interested in. So in the kind of Bristol region, in Bristol itself, north of Bristol, and in Glamorgan in South Wales, we have a lot of Carboniferous limestone rocks outcropping. The Mendip Hills, the Downs in Bristol, and many other examples around the kind of Bridgend kind of area of South Wales as well too. And in the Jurassic period, these represented small islands in a shallow tropical sea. And living on these islands were a whole range of different types of animals. And it represented in blue here, or purpley blue, represent deposits where we can find these animals. And these deposits are actually fissures which are occurring in these limestone outcrops. So many of these have been exposed at quarries. Most of these are exposed at quarries. Old quarries that were on the Downs, for example, that in Bristol that have now been filled in. There are a number of quarries in the Mendips and north of Bristol, and there are still active quarries working in the South Wales Glamorgan region as well. This is UNE, and you can see these quarries of the limestone is being quarried, but within those cracks in the limestone that we see, we've got these remains of animals that were around in the late Jurassic, the early Jurassic, scampering around that were then either dying and being washed in or were falling into these fissure deposits. Over time, their remains have been preserved as well too. So what we're getting here is a whole range of usually small animals. We've got early mammals, we've got also early representatives of things like lizards, we've got small animals which are on the line leading towards crocodiles as well too. And in Bristol as well, we have nearby, we've also got the remains of dinosaurs. This is an animal called Thecodontosaurus. This is in the foyer of the School of Earth Sciences in Bristol as you go in through the Wills Memorial Building. Again, a little model. This animal's not this high, not very big. Again, made by Bob Nichols, the same guy who did the Jurassic Scarborough reconstruction. So these quarries have been known to yield fossils for some time. Some colleagues of mine wrote a great paper recently about the history of these fissures. This is some images from the 1950s. It's a team from UCL, University College London, who were collecting these remains and using various different kinds of field gear as well. I think these are wonderful pictures. We've got the old car, very glamorous like field gear. Looks a lot nice weather. Not usually my experience of going to these places. Apart from perhaps the bottom left picture there. But also in the paper, it's a really fascinating story as well too about scientific rivalry between the team in London and another team in Cambridge that were also working on these same remains. And if anyone's interested, I can point you towards this paper. It's open access, so anyone can read it. But the value of the fossils that were coming out of here was realised. We're at the cusp of the origin of mammals, of lizards, of crocodiles, and so on. So we were particularly interested in these two kind of small, like proto-mammals that come from the fissures. And so for this, I was working with Pam, Pam Gill. Pam's an honorary researcher in Bristol. She did a PhD in Bristol. She lives on the Isle of Wight now. Pam's story is like a whole talk in itself, because she was actually started her PhD in the kind of 60s and 70s working on some of this fossil material, but the rivalry between the two groups meant that she was unable to access a lot of the specimens she needed for a PhD. So she packed in her PhD, she moved to Canada, she got married, she had 3 children, she became a school teacher. Over time, then she got divorced, she moved back to the UK, and in the early 2000s picked up her PhD again in Bristol. When the 2 protagonists of the argument had both passed away, the material was really available, and then she's taken it on from there and has remained fascinated all through that time in these tiny mammals. And I say tiny because these, you see the scale bar there, these things are like 20 centimeters, 20 millimeters, so 2 centimeters in size. They represent 2 different species, are both about the same size. The top one is called Morganucodon, which means Glamorgan tooth, after the area where it's found. The bottom one is called Cuneatherium, which is called Cune's beast. And Walter Cune, again, is a whole different talk in itself. He was a German living in the UK during the war. He was interred on the Isle of Wight, where he continued, the Isle of Man, sorry, where he continued to work on these fossil mammals. Then when the war ended, then went back to a career in paleontology, described some more of these and a whole host of other beasties as well too. Anyway, so we're back to working on these specimens. They're quite well known, but the problem is that we're at this interesting point now, where in the 50s, we thought all the mammals that were known in the Mesozoic, we thought small shrew-like things just scampering around. Since then, we've had a whole range of Jurassic and Cretaceous fossil mammals found in North America and in China mainly. Again, beautiful soft tissue preservation, which show that they're actually really diverse. And this image here, we've got examples of mammals from the Jurassic, then the Cretaceous, and then representatives from the modern day, which these fossil animals were probably quite similar to. So bizarrely, we've got animals in the Jurassic with soft tissue preservation with these huge beaver-shaped paddle-like tails. We've got animals which were kind of large, up to 10 kilo size in the Cretaceous, which looked like they might have been kind of terrestrial scavenger type animals, a bit like raccoons. We've got animals from the Jurassic, which have got forelimbs and claws, which are really similar in shape to digging animals like aardvarks and armadillos that are feeding on colonial insects. We've got animals that have got adaptations for tree climbing, and remarkably as well, the beautiful fossils from the Jurassic with soft tissue preservation of wing membranes, similar to flying squirrels and sugar gliders that we see in the modern world. So it's a real diversity of these mammals. Our little beasties, though, from the fissures sit just earlier in the time and in evolution to these amazing mammal fossils. And so what we were really interested in knowing was, were these early beasties also quite diverse as well too? You know, they were lumped as being these kind of general insectivores. Now, the problem is the stuff when you get them from the fissures, it looks like this. It's a big ball of jumbled up material, and it's all sort of stuck together. The good thing is, though, that these are all kind of infused. They're kind of in a limestoney or limey matrix, so you can actually either wash them or put a little bit of limescale remover in there, and then you can break up the fossils themselves. And the individual fossils, although they're jumbled up, are actually quite nicely preserved. The features are still quite clear. So this is where we used our X-rays again, but this time we've gone one step beyond the micro CT scanner that we have in our lab, and we're using synchrotron radiation instead. And this is because, not just for show, but because these fossils are super tiny now, and we really want to get really high resolution scans. And so what the synchrotron can do is actually using a very fast moving high energy beam that's been bent by magnets around these donut shaped rings, being able to create a very focused beam that gives us kind of a sub-micron level resolution, so less than 1,000th of a millimeter level resolution, which can pick up really detailed features in our 1,000th of a millimeter, of really detailed features in our fossils. So they're really quite amazing. We also look as well too, because we've spent quite a bit of time at these 2 facilities here. One is the Swiss light source, which is just outside Zurich. Very nice kind of environment there. And then the European Synchrotron Radiation Facility, which is in Grenoble as well. So you can see the Alps kind of nestling in the background. Fortunately, as well, at these facilities, we have people who know what they're doing to help us and make sure we don't damage the particle accelerator. It's actually remarkable the level of trust that they give you when you go into these places. We have all kinds of health and safety in our lab about not even being able to drink water when we're in front of the computers, and you might be able to make out there that Pam's got her cup of tea just nestled there. But they trust you, and they're built basically for people to come in, use the facilities, and then staff like Kathleen here in the pink jumper help us get really, really great results. And so we're able to use these for our fossil mammals. And so, as I said, they're often quite fragmentary, but often there are bits, there's so many bits that you can stitch bits and pieces together and try and get a 3D reconstruction of what the animals would look like. And we did this for Morganeucodon and Cuneiform, and we're able to get these 3D reconstructions of what we think the jaws look like stitching together 3 or 4 of our scans. So then we're able to use the same principle here that we used before as jaws, as levers, to work out the efficiency of the jaws. So we were working on the principle that these Morganeucodon jaws here they're much more kind of robust. They've got these big processes on the back where you would have muscles attaching and a big chunky jaw joint hinge here. The Cuneiform, it's missing a bit at the back, that's this grey area, but it's a lot thinner, a lot more what we call gracile. So we thought maybe that was a clue that these animals were feeding in slightly different ways. Their jaws are different shapes. So we looked at the muscle. Pam spent years looking at these things. She's got a good idea of where the muscles might sit. We use these estimates to work out the leverage of the jaws. And indeed, Morganeucodon's about 30% more efficient. So because these things are levers, we're working as efficiency here as being great at transferring force. So they're able to produce more forceful bites. The flip side of that are those Cuneiform's not as forceful, but it's actually able to move its jaws a bit faster. So you've got this trade-off between forceful or fast. So then what we did now was use an engineering method, which was alluded to at the start, called finite element analysis. And I'm not going to go into great detail about this. I'm happy to answer questions about this afterwards. I think some of you are engineers, you'll be familiar with this kind of method. It's used, we teach our students it. It's a standard method that's used to look at stress, strain, deformation. We're particularly using it for static structural performance, to look at where we see stress and strain and deformation in structures. These structures may be, golf balls, man-made machine parts. They could be, human teeth, jaws, hip joints, and so on. We're just using fossil bones instead in that context. And so we're using this method to look at the strength of our fossil jaws. And we find that Morganeucodon's jaws are about 50% stronger. So if we input a force here, which is a muscle force, and the finite element model then calculates the stress and strain within the jaws in response to that inputted force. And Morganeucodon has got much stronger jaws. So it's better at force transfer, and its jaws are stronger as well than Cuneiform's jaws are. So that's great. We could have just left it there and thought, well, actually, this is really good evidence that these animals are doing different things. The Morganeucodon's got bigger jaws, they're better at biting, they've got better force transfer, they're stronger. But we were really keen to work out what the animals were actually feeding on. And to do this, we used another method, which was tooth mark aware. So this is basically looking at high resolution maps of the enamel of the teeth to look at scratches and pits on the teeth. So when we eat things, particularly harder foodstuffs, they will for a while leave transient scratches and pits on our teeth. There's been some amazing studies on animals where they've been fed different types of things like hay with bits of grit in and things like that. And then you can take a latex peel of the teeth and look at the teeth and see there's different surface textures depending on what the animals were eating. So we worked with colleagues in Leicester who were able to use scanning electron microscopy to look at the surface of the teeth of our 2 fossil mammals, proto-mammals. And Morganeucodon's got loads of scratches all over its teeth. It's much rougher in texture than Cuneiform is. And that fits with our forceful biting, stronger jaws model. The methods that the guys in Leicester use actually allow us to create these digital elevation maps. So the hotter colours at higher peaks the cooler colours are lower troughs. And you can see that Morganucodon's got this really rough surface texture. But what we did from there was we thought, I wonder if there's actually living animals which have got similar tooth microwear signatures to our fossil mammals. And we had a student work on this project, and he worked with some colleagues of ours in Bristol who study bat ecology and got some specimens of UK bats. At the top, we've got horseshoe bats here. Horseshoe bats will typically eat, they prefer to eat things like beetles, of wasps, and things which are kind of slightly higher, more crunchy in texture. And then as a contrast to that, we have these long-eared brown bats here, which diet studies suggest they favour softer things like moths. And then we've got a common pipistrelle and a soprano pipistrelle, which we thought at the time were more like generalist feeders. They would just really feed on anything. So we got specimens of these bats. We took peels of the teeth and we looked at the microwear. And we found that if you look at them down a microscope, the horseshoe bat that eats more crunchy food has got similar tooth texture to what we see in Morganucodon. And the brown long-eared bat that eats softer things has got similar tooth texture to what we see in Cuneiform. Now the team in Leicester are able to basically quantify these different surface textures, and they use kind of standard surface textures from things like material science to like quantify peaks and troughs and orientations and things like that. And that allows us to build up a map then of the difference in tooth surface texture. So what you're seeing here is a plot where points that plot closer together have got similar tooth surface textures to each other. And we're able to draw boundaries pretty much distinctively around our different bats to show that they've got statistically different tooth surface textures. So at the far end here, we've got our long-eared bats eating soft food. And as we move along the scale here, it seems to correlate with a higher proportion of crunchy prey in the diet. And here are horseshoe bats here. So, and then what we can do is get our tooth surface textures from the fossil mammals and plot them on top. And Morganucodon are the triangles, and they plot here nearest to the horseshoe bats with the crunchy prey. And Cuneiform, these numbers represent different quarries and different fissures that these animals are from, but they tend to cluster similar to the softer food eaters here. And so we thought there's a few anomalies. You see there's a Morganucodon down here and there's a Cuneiform up here, but overall that data plots out quite distinctively. And so we thought that was pretty good evidence that actually these animals, and the jaws are not only working in different ways, but these animals are also feeding on different things. And we think that the Morganucodon is feeding on crunchier prey, Cuneiform feeding on softer prey. And indeed, in the fissures, we find remains of beetle carapaces, for example, which is what we've got here in this PAMA, this very nice reconstruction created here by an artist, John Sibick, and his Morganucodon homing in on a beetle. And in the fissures, we don't find moths, but we find scorpion flies, which are similar in terms of their probably a similar kind of softer prey item. So we've got Cuneiform here eating a nice soft prey item here. So what it was interesting and a great exercise for us because it allowed us to use lots of different methods. But I think what it shows is that you could bring in lots of different methods that help us really understand fossils in different and unique ways. And important in terms of mammals because it shows that actually even before mammals had evolved, these animals are still diversifying and occupying different niches, perhaps eating different food and eating them in different ways. So I will draw it to a close there. And thanks very much for your attention and for having me here to talk today. And I'm really happy to answer any questions. Thank you. Yeah.
Well, where do you think the rest of that gigantic plesiosaur is?
Right. So the question was, where is the rest of the gigantic plesiosaur? So we're pretty sure the rest of it is still in the cliff. Yeah. So on the TV show, there's a great feature that they keep returning to of them trying to excavate just the skull. So it was initially found because a guy, local fossil enthusiast collector, found the tip of the snout on the beach. So that had obviously fallen out the cliff first. And then the team went and looked to see if they could find the rest of the specimen. It was there in the cliff, but they had to dig into the cliff. And it's highly likely that there's a lot, given how well preserved the skull is, it's pretty likely there's a lot more of it, but it's going to go into the cliff by like 10 meters or so or more. And it's quite a long way down the cliff face too. So the farmer who owned the land was very happy to help them extract the skull, but he might be less happy for them to dig back 10, 15 meters away from the cliff. Yeah.
Is it possible for you to tell how often this biting action takes place, or how often these animals eat?
So the question was, is it possible to see how often the animals eat? Good question. So there's one way I would think about looking about that, would be thinking about the physiology of the animals. Okay, so whether they were warm or cold-blooded. And this is actually something that we're working on at the moment in terms of the origin of mammals. So we're warm-blooded. Mammals evolved from animals that were not warm-blooded, that were cold-blooded. And the question is, when on the lineage leading towards mammals did that actually happen? If you're warm-blooded, you need to eat a lot because you need to fuel your internal metabolism. If you're cold-blooded, you don't need to eat as lot. We know that from living animals. So it would depend on their metabolic physiology. And it was like, it probably wasn't a switch that went from warm to, from cold to warm. These animals were increasing their capacity to maintain their own, or generate their own body heat and then maintain it. So at that point, they will be eating more and more. So I think that will be how I would look at that question. In terms of looking at the jaws themselves, not, but I guess you could look at the teeth to see how warm the teeth were as well too. As we've got specimens for some of the Brazilian material that has lots of different ages. The teeth don't seem to be hugely warm, but these animals do seem to be living for quite a long time. So they're shrew size, but they might be living, as far as we can tell, up to 8 to 10 years. And we know that because the jaws themselves are still laying down bone seasonally. And so we're getting like a tree ring type structure in the bones themselves. And we're also getting that similar pattern in the cement that wedges the teeth into the jaws. It's kind of cool that you can pick that up as well too. So we can get an estimate of how old we think these animals are. So we think they're not fully warm-blooded yet because they're living for really long. They're shrew size, but they're living for like 8 years or so. That's not what we would imagine from a modern mammal that has a kind of live fast, die young kind of strategy for the tiny ones anyway. Yeah.
In sharks, you see the teeth falling out and being
Yeah.
replaced through the life. Do you see that in any of these fossils?
Yes, the question is in sharks that the teeth can fall out and are replaced during life. Do you see it in any of the fossils? Yes and no. So you see it in dinosaurs, they have a similar pattern of tooth replacement. It's the same as what you see in crocodiles as well too, where they're not as rapidly as sharks do. I mean, sharks, well, you find lots of shark teeth as fossils because they're constantly losing their teeth. And they've got 5 or 6 replacement teeth all ready to go in the jaw. In something like a dinosaur or a crocodile, you'd see 1, 2 at best in the jaw, but they do lose their teeth fairly frequently. Mammals like us, we obviously only have 2 sets of teeth. We have our milk teeth and we have our adult teeth. Animals that are on the lineage leading towards mammals like Moglucton and Cuneiform, we think are the first examples of animals that only have 2 types of teeth. Earlier forms, like our Brazilian specimens in our CT scans, have been brilliant because we can see 3rd replacement teeth. So we know they haven't evolved the true mammal condition yet of having only 2 types of teeth. So that's something we can see with the scans, that you could maybe see if you had a fossil, but you'd have to chop it up and make a thin section of it, and you'd have to hit exactly the right spot. I mean, these replacement teeth we see on the scans are tiny little things. No one's going to let us chop up these fossils. They're very precious.
Any more questions? Well, thank you for a delightful start to our season.
Absolutely.
I think and to warmly congratulate you on a totally absorbing talk this evening. My wife was trying to follow it on Zoom, and I apologize for my phone going ting because she said, we've had a complete power failure in the house.
Oh. I thought you were going to say the Zoom.
She's so she rushed off to our annex, but in fact, the whole of our internet's gone down, so something in the boots. But
So
anyway.
you'll have to tell her when you get home, everything.
Yes.
Oh, yeah.
Every bit, yeah.
Commit it to memory.
What I find absorbing this evening is that, you know, we all from childhood are just besotted by dinosaurs, and you've managed to make a career out of it. I think that's dramatic overkill. The other thing that sort of struck me too.
It probably is, yeah.
Yeah.
I agree.
The other thing that struck me this evening, I did my PhD 50 years ago, and I was trying to work on the movement of water through plants and leaves and how it was affected by pathology. And the way that you managed to integrate so many disciplines to tease out the story, I just thought was remarkable. And I wish I had those opportunities. And every time I come to these lectures, I find that technologies and things that have moved on that would answer the questions that I was trying to raise there. I was fascinated by your soft tissue, using X-rays to soft tissues with the potassium iodide, wasn't it?
Yeah.
That's right. I mean, I was trying to follow chelated lead through leaves.
Right, okay.
And the best thing that I could use at the time was I had to go to Southampton University Hospital's mammography unit, the breast cancer unit, because it's the only form of softest X-ray that we had available at that time.
Right, yeah.
And now I bet you could do it in I don't know. We shall see. Anyway, thank you so much for coming over this evening. It has been truly delightful, and I think we all owe Emily a vote.